Assalamu alaikum. Welcome, ladies and gentlemen, to tonight's keynote lecture. To introduce tonight's speaker will be Jean-Marie Basset, Center Director for the KAUST Catalysis Center. Jean-Marie?
Can you hear me? Okay. So tonight we are extremely happy, honored to have at KAUST during the web as a plenary lecturer Professor Hartmut Michel, Nobel Laureate in 1988, along with Johann Deisenhofer and Robert Huber. Professor Michel, you were born on July 18, 1948 in Louisbourg. I give your birth date because you have been one of the youngest Nobel laureates in all history of Nobel story, and you told me you've been the youngest for 15 years almost in chemistry. So this is really a very challenging situation. You are currently the director of the Molecular Membrane Biology Department at the Max Planck Institute of Biophysics in Frankfurt, but you are also the managing director of this center. You are also professor at the University of Frankfurt since 1989. You studied biochemistry at the University of Tübingen and then Munich from 1969 to 1975. I believe you chose Tübingen because it was the only university in Germany when you can start with biochemistry right at the beginning. Then you study for PhD at the University of Würzburg, where you got your PhD in 1977. After a postdoc at the University of Würzburg, you became group leader at the Max Planck Institute of Biochemistry in 1979, from 1979 to 1987. You got your habilitation at the Ludwig Maximilians University of Munich in 1986. Since 1987, you are the director of the Max Planck Institute of Biophysics in Frankfurt. Among all the prestigious awards that you received, let us mention that in 1986 you received the Gottfried Wilhelm Leibniz Prize from the Deutsche Forschungsgemeinschaft, which is the highest honor awarded by a German scientist. The Biophysics Prize of the American Physical Society, together with Deisenhofer. Then you get the Chemie doesn't, I'm sorry for my accent, doesn't stipendium of the Fond der Chemical Industry, the Otto Klum Prize for Chemistry, the Otto Bayer Prize, together with Deisenhofer, and now the Nobel Prize with Deisenhofer and Huber. Your contribution was inter alia, the success with the crystallization of membrane protein and the elucidation of the three-dimensional structure of the photosynthetic reaction center from the purple bacteria Rhodopseudomonas viridis. The bacteria surprisingly proved to be closely related to the photosystem II of the plants and algae. Your work was in several aspects groundbreaking. The size of the reaction center is enormous. The type of protein, and finally, the function of the protein. The size of the reaction center is enormous. The protein complex contains 4 proteins and 14 cofactors with a total number of 145,000 atoms. This was at the time the largest assembly which ever was crystallized and structurally characterized. Just imagine the difficulty to elucidate a crystal structure with 145,000 atoms in the asymmetric unit cell just by using diffraction patterns recorded on many piles of film. Number 2, the type of protein. The photosynthetic reaction center is a complex of membrane protein. You were the first to crystallize membrane protein, and still today there are only few groups in the world who can determine the atomic structure of membrane protein, such a structure typically seen in a Nature or Science publication. I'm sorry, I'm not responsible for this. The function of the protein, the structure of this center is highly related to the PS2 of plants and algae, which produces all the oxygen in the atmosphere and acquires the energy to fix 800 gigatons of CO2 every year and transform it into biomass. The purple bacteria photosynthetic center can actually absorb longer wavelengths light and is therefore more energy efficient than the one of algae. But you are here also because you have been a pioneer to start the fight against the food for energy. Your talk will be the nonsense of biofuels. It is fair to say that you are not only one of the smartest people in the world, but also one of the top experts in how plants turn sunlight into energy. To say that you are qualified to comment on biofuels, which is basically all about tuning sunlight into chemical energy via plants, is an understatement. Your views on this topic should carry a lot of weight. Professor Hartmut Michel, you have the floor.
Of course, I would like to start to thank the organizers for the invitation, for giving me the chance to present some controversial views here to this audience, and of course also Professor Basset for his introduction. And as already mentioned, I work at the Max Planck Institute of Biophysics in Frankfurt in Germany. It's a nice location, and actually there are about 200 people working in the institute, and they all work on membrane-bound proteins. So that's because we consider membrane proteins to be of great importance. And I would like to say a few words about that. So here you see a textbook figure of a cell of a higher organism, and everything you see is membranes. And membranes actually constitute a barrier. So signals have to cross the membrane, and also substances have to cross the membrane, and which is catalyzed by membrane-bound proteins. There are a few more functions connected to membrane-bound proteins. And I categorize the function of membrane proteins in 4 categories. And first, I mentioned already the specific transport of substances across the membrane. And you have active transport, you have passive transport. And passive transport by channels, active transport and carbon transport by transporters. It's not so evident that most biological electron transfer is catalyzed by membrane proteins, and it's mainly in photosynthesis, but also in cellular respiration. Most important for medicine is that membrane proteins, as I already shortly mentioned, act as signal receptors, and the membrane proteins transduce the signal across the membrane and amplify it, and some of the membrane proteins are enzymes, preferentially when the substrate is hydrophobic. What's very important for the applied science is that 80% of all marketed drugs act on membrane proteins, primarily on signal receptors, but also on transporters. And that's why we do some work on that. My main scientific interest at present is respiratory chain. That's the mitochondrial respiratory chain. You may know that, and this is now also the membrane protein structure is close to being complete, and these complexes are integrated into the membrane of the inner mitochondrial membrane, and the job is to oxidize NADH, which has hydrogen bound NADH, and the electron is then transferred along this line towards the cytochrome C oxidase. Cytochrome C oxidase generates water, and protons are pumped across the membrane, and membranes act like capacitors, so you can build up electric voltages across the membranes, and of course you also get gradients of substances across the membrane. And the pH gradient then the electrochemical proton gradient then drives protons through that machinery, the ATP synthase, and leading to the rotation here of this blue part here of the ATP synthase. And the rotation is covered to the synthesis of ATP from ADP and inorganic phosphate. And we like to understand at least how this machinery works. And I should say that this machinery, this protein, was actually discovered already in 1886, but we still do not work how it functions. So this is still a scientific channel. But today I'm not talking about this scientific stuff. I'm more talking about thing of more general interest to the public about efficiency of photosynthesis and the consequences for humanity. Start off with here, you're all aware that we have global warming, and there's no doubt that global warming exists. And for the moment we are up here, but we have to say that in the last 15 years there was no further increase of the global temperature observed, but we can expect that the temperature goes up further. And this is correlated to the carbon dioxide concentrations in some way. You see here the carbon dioxide concentration measured in the atmosphere, and for the moment we are reaching 380 ppm, and this is correlated also, of course, mainly to the burning of fossil fuel. That's the total fossil fuel burning, but much of that is absorbed in the water, so not everything which is carbon dioxide which is produced appears in the atmosphere. We also have this nice correlation over the last, I think, 900,000 years now, that we have variations in the CO2 concentration, and this is parallel towards the temperature change you see here, and we have this occurring about every 110,000 years. This is called the Ice Age cycle, and for the moment we are in a warm age in Europe, and but one has to say here that the correlation doesn't say that the carbon dioxide concentration leads to the temperature change. On the contrary, we have to see that there is a different reason for that. But at least it gave rise to the consideration that the increase in carbon dioxide and other greenhouse gases, primarily methane, but also nitrogen oxides, cause the observed temperature rise with the global warming. And looking back, the temperature rise precedes the rise in carbon dioxide concentration. So looking back to the ice age cycles, we cannot say that it's the carbon dioxide concentration which gives rise to that. And the reason for the correlation is that the temperature rise, due to other causes, simulates the activity of bacteria, aerobic bacteria, leading to carbon dioxide release more than it helps photosynthesis. On the contrary, for carbon dioxide fixation gets disadvantaged when due to the increased temperature, because the solubility of carbon dioxide is lower, which is a limiting factor in photosynthesis. I will come back to that fact later on. But we have still pretty good evidence that global warming is caused by greenhouse gases, and primarily based on calculation simulations. That's based on the infrared radiation transfer theory, and already worked out by Svante Arrhenius in 1896. The best piece of experimental evidence that we have this global warming due to greenhouse gases is that the higher atmosphere, the stratosphere, is cooling. That's observed by measurements for satellites, and it's cooled down because due to the increased concentration of the greenhouse gases, the higher atmospheres gets less infrared radiation from the surface of the Earth. This is the best evidence that actually greenhouse gases cause the global warming. Of course, we have now the predictions from the various simulations, and the expectation is that until the end of the century, we will get an increase between 2.5 and 6%. Simulation is different. What is not very clear is the influence of water. Water is the most dramatic greenhouse gas, and the influence of the clouds cannot be well represented for the moment, so there is some uncertainty to it. Now, how we can remedy the whole situation, and because, as you all know, fossil fuels, coal, petroleum, natural gas are derived from photosynthesis. And in photosynthesis, plants fix carbon dioxide from the atmosphere. Question then is, can plants be used to produce biofuels? So we have two intentions. We could solve the energy problem of mankind, and at the same time, we could reduce global warming. Then, of course, I turn towards description of photosynthesis. You see here, that's a leaf. That is where most of the photosynthesis occurs. And some basic facts. Photosynthesis has to be split in the light reactions. This means the absorption of light leads to the creation of chemical energy, redox energy, fixed hydrogen, NADPH, and ATP as an energy currency. And oxygen is a waste product in photosynthesis occurring in the atmosphere, first time about 3 billion years ago. But it took another more than half a billion years until the atmosphere became highly oxygenic. Dark reactions is the second point. These are the synthesis reactions. The stored redox energy and ATP are used to take carbon dioxide out of the air and convert it as sugar. And schematically, we have here the light of the sun is absorbed by the plants, by the algae, and leads to the synthesis of ATP and NADPH, and releasing oxygen into the atmosphere. And ATP and NADPH are used to reduce carbon dioxide and convert it into sugar in the Kelvin cycle. I'm now going to discuss what's going on. In the first step in photosynthesis is the absorption of light by the carotenates and chlorophylls of the light harvesting antenna. That's an antenna protein of a purple photosynthetic bacterium, different one. You see here these rods are actually helices, and it has a nice symmetric shape in this bacteria. And you see here the chlorophylls form two rings. We also have carotenoids, which have a photoprotective function, but also a light harvesting function in these systems. And they transfer the energy to the reaction center. And in the reaction center, we have the charge separation across the membranes. Surface would be here. That's integrated into the membrane. And the energy is stored in the form of the reduced electron acceptor and in the form of the electric voltage across the membrane. And this reaction happens in this part here. And here I show you the photosynthetic pigments. So here we have so-called special pair. That is the primary electron donor. If this gets excited either by energy transfer from the light harvesting complexes or by direct absorption of light, the electron is transferred here, and to here, and then to here. And it's a symmetric structure, but only this pathway is used for electron transfer across the membrane. So we get here reduction of the quinone, and we get the electric voltage across the membrane in this step. And all the reaction centers in plants have a similar central architecture, and what you can see here is a view onto the photosystem, one reaction center of the plant, and here in the heart we have that what corresponds to the bacterial reaction center. There are some modifications, but the basic symmetric feature is there. We have a non-covalent chlorophyll dimer as a primary electron donor. And when you have a closer look here into this plant system, you see here the special pair. It's now, unfortunately, opposite orientation. You have here the chain of chlorophylls and pheophytins up to the quinone, but then in the plant photosystem 1, we have here as an iron sulfur protein, iron sulfur cluster here as the electron acceptor from this plant photosynthesis. Looking now into the electron flow in the chloroplast and cyanobacteria, first we have here photosystem 2. Photosystem 2 gets excited, and this leads to accumulation of positive charges here. And this then leads via manganese cluster to the splitting of water and release of oxygen. The electrons are here, and they end up in another membrane protein complex, which converts the electrons back across the membrane with some energy conversation. Then we have that's the B6F complex here, reducing plastocyanine. Plastocyanine then reduces and photooxidizes photosystem 1 reaction center. I just showed you the structure. And the electron then is here transferred to ferredoxin iron sulfur proteins, and then at the end used to reduce NADP to NADPH. So it picks up protons here As a result, we get a pH gradient and electric voltage across the membrane, and this is what is used by the ATP synthase for generating ATP in a similar way as I outlined in the respiration. So the machinery, the components are more or less identical here, ATP synthase in photosynthesis and in cellular respiration. Now let's talk about efficiency of the process. And it's pretty clear the quantum yield is very high. But this doesn't mean that the energy yield is high, because less than half of the sunlight reaching the Earth is photosynthetically active. It's about 47% of the sunlight can be absorbed by the plants and used to drive photosynthesis. As I mentioned already, quantum yield is high. That means that each photon absorbed leads to electron transfer across the photosynthetic membrane, but this does not mean that the energy yield is high. And most of the light energy is already lost in the primary light reaction. 8 photons, in theory, are used to rise the energy of 4 electrons by 1.2 electron volts each, meaning that between 19 and 33% of the energy of the absorbed photons is only stored in the form of NADPH. So here we have the level where the electrons start off in water and then they are transferred to NADPH, and the rest of that actually is the rest of the energy actually is useless, cannot be used in further reaction, is lost. And experimentally, it's not only 8, it's actually all measurements show that you need 9.4 photons to reduce 2 molecules of NADP plus to NADPH. And if you consider that only 47% related to energy of the sunlight is photosynthetically active, we have to conclude that about 12% must be the absolute maximal efficiency of photosynthesis. That's already in the light reaction. And this value, however, is substantially reduced further by the inhibition of photosynthesis at high light intensities, by damage at high light intensities, and by the inefficiency of the carbon dioxide fixation. What happens in photosystem 2 reaction center, similar as it happens in the photosystem 1 reaction center, we have the primary electron donor, that special pair. The electron is transferred to a chlorophyll, then to a pheophytin, then to a first quinone, then to a second quinone. And always the following step is slower than the previous one. And if an electron then comes in before the electron has to transfer from pheophytin to the QA, it cannot reduce the pheophytin any further, and it has to fall back onto the donor, and this can lead to triplet states and leading to reactive oxygen species and oxidative damages. And Planck can repair that, and the central protein called D1 of photosystem 2 is exchanged in the plants 3 times per hour. So the plants manage to cope with the photo damage in that way. But we also have the problem that we have inhibition of photosynthesis at high light intensities. And this is caused by the fact that under normal sunlight, the photosynthesis is oversaturated. We get too much energy from the light harvesting complex onto the reaction center, and the reaction center cannot cope with the electrons. And already at 15 to 20% of the full sunlight, the photosynthesis is saturated. So we lose more than 80% of that in the synthesis step of the absorbed energy. We cannot make use of that. We have further limitations. One is the lack of water. Photosynthesis depends very much on the availability of water. And the other point is the inefficient fixation of carbon dioxide. And upon the loss of water and the dehydration, the stomata actually at the underside of the leaves close, and this reduces the loss of water, but also the availability of carbon dioxide becomes limited because it cannot be taken up by the stomata, and photosynthesis stops during much of the day when the sunlight is shining, fully shining. And the last weak point, major weak point, is the enzyme Rubisco. Rubisco stands for ribulose 1,5-bisphosphate carboxylase, and this enzyme cannot discriminate accurately enough between carbon dioxide and oxygen. So the reaction is like this, that we have here this sugar ribulose 1,5-biphosphate, and the Rubisco enzyme takes carbon dioxide and puts it into this 3,5 sugar and splits the sugar into here. And here the problem is that this enzyme does not discriminate between carbon dioxide and oxygen good enough. It discriminates by a factor of about 80 But considering that the carbon dioxide concentration in the atmosphere is much, much lower than the oxygen concentration, you get an incorporation of the oxygen in about a third to the fifth reaction cycle into the sugar. And the incorrect product is 2-phosphoglycolate, which has to be degraded, and this uses a lot of energy. And it's calculated that the energy of about 1/3 of the absorbed photons is used for this purpose. So that is an enormous problem. The other point is, I could mention that Rubisco is a very sluggish enzyme, probably the slowest enzyme known, and it catalyzes only 3.3 reaction cycles per second. And this slowness is compensated by the synthesis of enormous amounts, and up to 50% of the soluble proteins in the plants consist of Rubisco, and it's therefore by far the most abundant protein on Earth. There are people believing that it cannot be improved. This discrimination between carbon dioxide and oxygen cannot be improved. And if you would increase that discrimination, the enzyme would become even more sluggish. But this has to be investigated. Structure of Rubisco is known since about 20 years. Look, very nice proteins in spinach. It is a symmetric octameric protein. And so now coming to the summary of that, for normal C3 plants, 4.5% is considered to be the upper limit of the efficiency of photosynthesis. But in reality, this value is not reached. On an average, less than 1% of the energy of the sunlight is stored in the form of biomass. So that is not a very good thing. Let's present a few examples. So I'll show you here an example of potato. And potato contain about 0.5% of the energy of the sunlight shining onto the field. But we have to consider that 60% of the energy in the harvested potatoes comes from fossil fuels, indirectly via fertilizers, via tractor fuels, via fungicides and insecticide sprays, and not from the direct utilization of sunlight. So it's about 0.2% of the sun's energy which is net in the potato. We can go further on with biogas. Biogas is produced by methanogenic microorganism, archaea, from biomass, and it contains about 60% methane. The rest is mainly carbon dioxide. And how this is done in Germany is done mainly by farmers or by people buying the waste products from farming from the farmers. Is hosted into fermenters, and the fermenters, you have the archaea there, and they convert the sugar and the carbohydrates into methane. Primarily in Germany, it's growth of maize, and nowadays 20% of the German agricultural land is already used for the growth of maize, of maize corn, for the production of biogas. And as a result of that, Germany became a net importer of wheat. We were an exporter of maybe 3 to 5 billion tons of wheat 5 years ago, but in the last 2 years we became an importer of wheat in the same amount. So we have already changes in economy due to the generation of biogas from the agricultural area. And we can go on with calculations, and we can get about 4,600 cubic meters of methane per hectare of land, and this contains an energy of about 46,000 kilowatt hours. And this would be converted to 70,000 kilowatt hours electric energy, and if you calculate this per square meter, it's 1.7 kilowatt hour, and here this results about 0.2 watt per square meter on a continuous basis. And if we have a look, what is the average energy of the sunlight reaching the Earth, then it's here in Germany, in South Germany, it may be 150 watts per square meter. Here in Saudi Arabia, it's about 300 to 350 watts per square meter. So it's a factor 2 to 2.5, which is the difference between Germany and Saudi Arabia in that respect. So I mentioned already 150 watts per square meter. This converts into 0.2 watt into electric energy, and this means that only 0.13% of the energy of sunlight has been converted into electric energy. And we have not considered that we had to put in around 30% of the energy in the form of fertilizers for tractor fuel to produce the biogas. So we are below 0.1% efficiency generating electricity in that way. If we then go on and consider, are we able to generate all the electric energy consumed in Germany by biogas, then we can clearly say no, because we would require 720,000 square kilometers, and the entire area of Germany is only about 350,000 square kilometers. The agricultural area, about 150,000 square kilometers. There's no way to produce all the electric energy used in Germany via biogas. Next example I can present is bioethanol. In Europe, that's produced from sugar beet or from wheat. In the United States, from corn. And it has an energy content, the ethanol, slightly higher than the biogas. But the energy input is pretty high in Europe and in the United States because much of energy has to be invested into the growth of the plants, the harvest, and into the concentration of the alcohol to 99% by distillation and other chemical processes. And it depends where you get the energy from the concentrating the alcohol, whether it is a net production or net loss of carbon dioxide. There's practically no effect when petrol is used for providing the energy input. So that is not very efficient. Situation is slightly better if you consider sugarcane. That's the way Brazil does it. And it saves carbon dioxide primarily because you don't plow the field every year and plant the plants freshly as we do it in the United States and in Europe. And the squeezed stems from the sugarcane are dried and burned for distillation. And the energy input is only about 1/9 of the energy content of the ethanol produced. But if you do similar calculations, you know what the energy yield of alcohol and what is the energy of the sunlight shining into the field in Brazil, then you easily can calculate that even Brazilian bioethanol contains less than 0.2% of the energy of the sunlight which has fallen onto the sugar cane plantation. So it's very low. Next generation of biofuels, which is considered, is the biomass to liquid transfer, BTL, and because conventional present-day techniques like bioethanol production from sugarcane and sugar beets or biodiesel production from rapeseeds use only parts of the plants. And the future BTL production is supposed to use the entire plant, which is gasified or converted enzymatically, and less land is required to get the same amount of fuel. And for synthesis, you use syngas via the Fischer-Tropsch process, and this diesel is called FT diesel or Fischer-Tropsch diesel or sun diesel sometimes. And if we then go on and do similar calculations, the yield, which has been claimed by some companies, is that you get about 1 liter of this diesel from 4 kilograms of wood. And had also been claimed that that you get about 3,000 to 4,000 kilograms of that diesel per hectare. But recent tests by European Union actually indicated that you get considerably lower values of about 890 to 2,300 kilograms per hectare. And in order to get the high yields, you have to add hydrogen gas. And hydrogen gas is produced, of course, from fossil fuels. So that is also not very efficient. And if we have a look whether we can get the German gasoline or diesel by that process, you can clearly say no, because the entire Germany area would be required to grow poplars or miscanthus in order to supply the present consumption of gasoline and diesel for cars and trucks. And these are the trees which are considered. That is poplar. That's a tree in Germany, growing very fast, has pretty high photosynthetic yield. And also this grass is also used also in the United States for getting biomass converted into the biodiesel. In the tropics, you have here the oil palm, and this comes from the fruit of the oil palm, and it has a pretty good yield, 5,000 to 6,000 liters. And the major problem there is that the rainforests are cleared, primarily in Indonesia, Malaysia, but also nowadays in South Africa, in Africa and in South America. And but these rainforests, they grow quite often on peat, and this organic matter in the soil will be oxidized by bacteria after clearing the forest. And there are indications saying that the carbon dioxide saving using palm oil as biofuels will only be compensated in 430 years. in particular, in order to save the tropical rainforest in Southeast Asia, but also, as I said, in South America and Africa, the import of palm oil should not be used in Europe. We need actually rainforest as a source for chemicals, and lots of the pharmaceutical basic compounds are derived from compounds from the forest. And if we clear that, actually we will lose many compounds which will not be discovered if we clear one in the forest. Apart from that, we will certainly have climate changes if we clear the rainforest and replace it by oil palm plantations. You can compare the crop efficiencies for biodiesel production, and this I took this from this web page, from the solar-biofuels.org, and they compare here the various yields. Soybean is the least efficient, and palm oil is the highest. They claim that algae are much better. And even if you have a look here at the algae, the high claim, and if you now do here the calculations, you end up with a value which is by a factor of 3 above the theoretical limit of photosynthesis. So this is what is the major problem, that these data are not accurate and they are grossly exaggerated. Of course, we can discuss what is the advantage, disadvantage of algae, and they have positive arguments. There's no lack of water, and all cells are photosynthetically active. The difference from the plants, no roots, and there's some contrast. And this is Rubisco of green algae is worse than that of land plants. And but you can, in closed systems, you can add carbon dioxide and get a compensation for it. And for the inhibition is similar as in land plants. There's no difference with photoinhibition in land plants and in algae. So there are already some kind of demonstration plants. You see here, that's in Arizona in the United States. There's a conventional power station. Carbon dioxide is produced, and that is pumped here onto the algae growing fields. And this is then considered to be used for biofuel production. I don't think that biofuel production from algae will ever be commercially competitive. I would think the yield can be by a factor of 3 to 2 higher than with land plants. But I would think the value of this using algae will be in producing more valuable compounds than simple biofuel, primarily vitamins and chemicals which can be converted into building blocks for organic synthesis of a higher value. And however, my vision with that is that we, for the synthesis, which of course is also important for farmers growing food, that we can improve the carbon dioxide fixation, the Rubisco enzyme, by genetic engineering and selection techniques. I would estimate that we can increase the overall yield of photosynthesis by 50 to 100%. Second point would be one can try to expand the wavelength range used by the plants. We could introduce light harvesting systems absorbing UV light, and also green light and infrared light, but the leaves should be black. And this is not, say, a very positive prospect if we have that. And most interesting, in my opinion, is we can reduce photoinhibition at high light intensities by reducing the size and number of light harvesting complexes and increasing the percentage of the reaction centers. This really would be a big advantage. But still, we have the problem, water availability will be crucial. And there are reports that in order to produce 1 liter of biodiesel from rape seeds, you need 40,000 liters of water required. And so this is, of course, a very bad message. And if you would use irrigation for producing the plant material for biodiesel, it would be very, very bad. My final point against the biofuels here is if we try to reduce carbon dioxide release, it would be much better to grow poplars on the land used for biofuel production and convert the biomass into coal. This can be done by a process called hydrothermal carbonization. It's an energy yielding process. You heat the biomass in water to up to 160 degrees, and you convert all the carbon dioxide to coal. And so it's still there, no carbon dioxide released, and this would save 2.7 kilograms of carbon dioxide per square meter. If you produce biofuels, you maximally can save 0.1 kilogram of carbon dioxide per square meter. So the difference here is by a factor of 10, and that is why that's a very strong argument against the production of biofuels. If you have biomass available, it is much better to use that for direct energy conversion by burning, or to use it in power stations for generating electricity instead of putting energy into the conversion of the biomass into biofuels. Of course, we have to look for the alternatives. The alternative comes into mind is photovoltaic cells, and you can buy from the shelf photovoltaic cells which have an energy yield of 20% of the sunlight's energy, which is not reachable by photosynthesis. And now, alternative to that is solar thermal use. You have here mirrors, and the mirrors, that is one version of that. The mirrors can focus the sunlight into tubes. This is filled by a liquid. The liquid can be heated up to 400 degrees centigrade, and this is then used for generating steam and electricity. And some people say that the efficiency of that process is in the order of 10%. I don't believe that. I think it should be slightly lower. And it also can produce electricity in the night, because you can store the heated liquid and use it for generating electric energy in the night when the sun is not shining. Okay, we can now go on. What would be the space requirement for solar cells, providing all electric energy for the world, for Europe and for Germany? And this is for electric energy. That is a square like this. For Europe, it's a square like this. And for Germany, it would be here a square of 50 times 50 kilometers. And this would provide all the energy for Germany up here from the Sahara. And there's actually an initiative trying to do that. It's called Desertec. And there are already some of the solar thermal plants being constructed here in Morocco, some in Tunisia, but also some in Egypt, and which at the end could provide electric energy to Germany. My vision here would be that if we would be able to have superconducting electricity cables, we could transport electric energy without losses across the globe. Then 3 or 4 big photovoltaic fields, maybe one in North Africa, or here in Saudi Arabia, one in China, or one in Australia, one in Mexico, and we would connect these fields by superconducting cables. We would have sufficient supply of all electric power which mankind needs. There would be no need to store the energy, because if it's night if it's night here in Africa or in Saudi Arabia, then it would be day in Mexico and Australia, and we would have always energy created from sunlight. But even without the superconducting cables, we can transport electric energy from the Sahara to Central Europe by using high voltage direct current cables, and this is currently done in the Desertec initiatives. But how we go on with driving cars? We have here a car, Tesla, an American car, and the car uses 80% of the energy stored in the electric batteries for propulsion. If we go for a combustion engine power car, this uses only 20 to 30% of the energy of the gasoline to drive the wheels. So here we have an advantage of a factor of 3 to 4 for the electric propulsion of cars compared to the combustion energy for driving cars. And for that, one has really to postulate that the cars should be driven by electric engine using the electric energy directly derived from sunlight. If we consider that only 30% of the fuel energy on combustion engines is used for propulsion, and biofuels contain less than 0.2% of the energy of the sunlight. On the other hand, we have the combination photovoltaic cells, electric battery, electric motor, and this combination uses the energy of the sunlight at least by a factor of 500 better than the combination biomass, biofuels, combustion engine. The limitation is clearly the energy density of the batteries. We need more powerful batteries. And already in the lab, there are novel tin lithium sulfur batteries which can store 10 times more energy per weight than the present day's lithium batteries. And they have to be developed and they have to be engineered to be safe. And then so such kind of electric car would have the same range for driving than a combustion engine driven car. For transition time, we could use hybrid engine, which is primarily powered by batteries. Summarizing that, production of biofuels from various kinds of biomass is a very inefficient land use, and much fossil energy has to be put into the production of the biomass and its conversion into biofuels. And if you have biomass, it's preferable to use the biomass for heating or electricity conversion in power plants, replacing fossil fuels. And we have to use solar energy to generate electricity either via solar thermal power plants or by photovoltaic cells. And cars have to be driven by electric batteries and electric motors. Of course, without that, jets can be driven by electric batteries. Still, we still need kerosene to produce, and this I think the chemists can do via various ways. And the outlook, the renewable energy sources, sun and wind, cannot provide electric energy continuously, and we have to store the energy when the energy is in excess. Actually, we have that already in Germany, where we have lots of photovoltaic cells installed, and at good sunny days, 70% of all the electric energy is provided by photovoltaic cells, and still we have the coal-driven, coal-powered power stations running, and we produce much more electric energy than we actually can consume. So we have to find ways to store the energy. Of course, there are physical means. We can pump water uphill. It's not very suited in Germany because we don't have so many hills. We can press air into cavities, which still have to develop further and to reduce the backflow of the air for generating electricity. We have chemical conversions. We have to electrolyze water, generate hydrogen gas. Then you can produce electricity again from the hydrogen using fuel cells or by burning the hydrogen in conventional power stations. And actually, this is already done. The first plant operating in Germany in the Frankfurt area generates hydrogen by electrolysis of water. But this is now used actually to fill the pipes used for natural gas. And the natural gas in Frankfurt now contains hydrogen produced by electrolysis of water. And we have the conventional means. We can produce methane, methanol from hydrogen and carbon dioxide. And we can of course go up if we have methanol. Methanol we can go up and make longer chain lengths. And they're also providing transportation fuels. And I was very happy to see today all the efforts in the catalysis center to improve the biosynthesis of long-chain hydrocarbons for that purpose. I think there's a chance that we will have enough energy even if the fossil fuels has been run out. And with that message, I would like to close and thank you very much for your attention.
So please lift your hand if you want to ask questions. Here? Please ask for a microphone.
Hello. Yes. Thank you for your speech. But I have a concern specifically regarding the biogas. Why you have included biogas production in photosynthesis efficiency, while photosynthesis is a natural process that light is considered as the energy source, while biogas production is a design process where carbon is the energy and carbon source. And the second part, if we need to study this, the photosynthesis efficiency in nature, do we need to consider the growth rate of microbial community that involved in biogas production in photosynthesis efficiency?
Of course, it's no doubt that biogas, or the methane, is derived from biomass. Because what you add into the fermentation plants are either the residuals from farming or maize or corn leaves, in particular, specifically used for the production of biogas in this fermentation plants. There is biogas therefore is biofuel, and of course in Germany we also use lots of methane for driving cars instead of diesel, and this is another way of using methane. With respect to photosynthesis, I can simply give you the figures, and there's no doubt that these figures are correct, and that the overall efficiency of converting or storing sun energy in the biomass and the biofuel is extremely low. And you should do everything not to convert going from the electric way to the chemical way, because converting the biomass into biofuels is always lots of input of energy.
I'm here. I have some questions about the improvement, the potential of the improvement of photosynthesis and photoinhibition. So if you look into different, into the evolution, you mentioned, for example, that algae have a much less efficient
It depends.
Rubisco.
I said the green algae. Actually, what turned out is that the red algae have a much more efficient Rubisco. Actually, they discriminate by a factor of about 300
Yeah.
between
So
carbon dioxide and oxygen. And this, for me, is an indication that Rubisco in the plants also can be increased.
Right.
And I said if we increase Rubisco, probably I would think that 50 to 100% of the overall photosynthetic yield we will gain. But if I talk to specialists in Rubisco, they tell you clearly there's no way to improve Rubisco. When you make Rubisco better with respect to discrimination, you reduce the overall turnover. And you have to compensate for that by making more Rubisco, and there will be no net effect on the overall carbon dioxide fixation. But the red algae do it. And one has really to, the red algae can do it, so I don't think that is a compelling argument that you cannot improve Rubisco even further. You have to invest and not believe that it cannot be done, and just do the experiment and try, and I think that somebody will be successful. Of course, you also can try to take the Rubisco from the red algae and to put it into the plant system to optimize temperature dependencies. Will be some work to be done, and you will up with a much better plant, with a much better carbon dioxide fixing plant.
But that means actually that there is scope for improvement, mainly also by looking into the biodiversity on the planet, right?
Sure.
In both ways. I think specifically maybe for the hot zones with a high light intensity, as you said, one could look actually to improve the photoinhibition side.
Yeah. With respect to photoinhibition, of course, it's a little bit difficult because photosynthesis in general is optimized for low light conditions. Yeah? And it's with 5 to 10% of sunlight, it runs maximally. But if further increase doesn't help you, but it's mainly because we have too much light harvesting antenna, not enough reaction centers. And for that, the way would be to reduce the number of light harvesting systems and to increase the number of reaction centers synthesized. And thereby we would get much higher efficiency of photosynthesis by that. And there are actually ways to try that. Also, other examples which I didn't mention is that you try to convert C3 plants into C4 plants. C4 plants have a little bit higher photosynthetic efficiency because they have a mechanism concentrating carbon dioxide for Rubisco, so the discrimination, the lack of discrimination between oxygen and carbon dioxide doesn't have that dominant role as it has in the C3 plants.
But C4 plants are only advantageous under high light conditions, and given the incredible importance of photosynthesis and the processes you've been studying for so long, given the long evolutionary history and the long amount of high selection pressures on that, don't you think that those sorts of processes are likely to have already been optimized?
No, the point is if a single mutation or two mutations are always detrimentous, and you would need 4 mutations at the same time to improve, nature cannot do it. And if by design you could do it.
Thank you for the talk. I would like to ask your comment on last part of your presentation about the CO2 conversion. So how do you see the future for the CO2 conversion to biofuels, either methane or methanol? Do you see that in near future it would be a kind of commercially available reaction? I would like to have your comment on that.
I would think the basics is known, and there are still lots of development with respect to technology development has to be done. You have to develop catalysts being more efficient, so you reduce the energy input used there. Generating hydrogen is already pretty efficient. But of course then the combination, using hydrogen and carbon dioxide to produce methane or methanol, and then go up to higher substances is more difficult, but I think it can be solved. Whether economically is difficult to say, whether it's economically viable. But as soon as there is no longer any natural fossil fuel available. Price doesn't count.
Excuse me. The question relates to the increasing the absorption capacity at the ultraviolet radiation region and also the far region. And my question relates to how do you think the reaction centers will process this absorbed energy in excess in the case of photoinhibition or at UV light irradiation?
Well, if there's too much light, it's converted into heat. Huh? If you have, it's lost in heat. And in particular, but also, of course, if you have the process that you have a double excitation of the primary electron donor, and you have already reduced next electron acceptors, you generate triplet states. Triplet states get singulate oxygen also there, and, which is the damaging case. And the plant handles it by repairing the photosynthetic reaction center. Technological process, I don't think we can do that. But in general, my opinion, we can do that. Does this answer your question?
Yeah, but I would like to know how practical it is to increase the efficiency by changing the absorption of these two extreme ends.
Okay, that is in my opinion, I don't think it does matter. The green light, we could try, for instance, we could add some system from algae. Either from red algae or from cyanobacteria, some phycobilisomes to add something where they have some absorptions of the green light occurring. And the UV light should be able, by simply reducing the number of conjugated double bonds in the carotenoids. But infrared light, I don't see practical that we can use infrared light. Of course, bacteria do that, but then the energy transfer would be problematic, because we have to have an energy transfer uphill, which is sometimes possible in some kind of bacteria, where the light harvesting antenna absorb light at longer wavelengths than at the special pair of the reaction center. There it can happen, but in the plants, I think the difference is too big. So that is also minor. It's only a minor point of improvement, in my opinion. No? Primarily, I think UV light could be used, and also green light could be used. Infrared light, not so likely.
Okay, thank you for your lecture. It was very interesting for me. But next February, in a few months, I will start here a lecture on the importance of biofuels. So I cannot agree with all of your conclusions. That I would like to use this opportunity to make some advertisement for this course. Anyway, I agree with some of your conclusions. I know there are many projects in biofuels which are total nonsense. They're spending a lot, especially some of the large algae projects. But there are other modern biofuel plants which are very effective with a very positive energy balance. And one can prove that the input of fossil fuels is only, let's say, 10 or 15% compared to the output of energy.
That's the bioethanol in Brazil, no doubt.
So your lecture title was too radical for me, the nonsense of biofuels. Yeah. It's not, some is nonsense, but some really makes sense.
If respect what you want to do, if you want to reduce carbon dioxide concentration to reduce global warming, it's nonsense, because converting the plant mass into carbon is reducing 10 times more, is consuming 10 times more carbon dioxide, reducing the carbon dioxide by a factor of 10 compared to biofuel production. If you go for energy, we still have a little bit a niche for biofuels, of course, apart from Brazil. But you cannot compare Brazil, because Brazil has lots of land, not much population, but you cannot biofuel that way in India or in China.
Good, thanks.
I have a comment, and I'm here. Yep. Thank you for your talk. I have a comment and would like to hear your opinion on it. So I think one of the big problems is the car industry. So if you look at the history of the car, it's over 100 years old. And for example, the Ford T, that was the first commercially produced car in 1909 or something, was already running with ethanol. Had a combustion engine, looked more or less like my Kia that is in my driveway. So if you compare the evolution and the technology in cars, especially in the combustion engines, with, for example, the change in computers over 50 years or 30 years, I think it's really minor compared to other technological advances that we made. And I think one reason for this is that the car industry is very hesitant to implement new things because they are producing so many cars, it's working well, they make a lot of money. So don't you think that a lot more effort has to be done on the side of the car industry rather than on the research side on biofuels or solar cells?
There's lots of pressure onto the car industry, but you know that European legislation now gives you an upper limit for the carbon dioxide release per vehicle on average. Yeah? And this puts the companies under pressure to produce more efficient engines, and also to reduce, of course, to make the energy loss due to the friction. To reduce the energy loss due to the friction, just the English word escaped, so it slipped out of my mind.
Deutsch funktioniert auch für mich.
Okay, good. Also der Luftwiderstand, to reduce the Luftwiderstand. Yeah?
But in general, wouldn't you say, if you look back in history 100 years, that the car should have evolved much faster than it did?
It could have evolved faster, because there was no need actually to. The gasoline price was so cheap, and there was nobody was aware of the environmental problem. People didn't take care. But for the moment, actually, I would think the pressure in the industry is very, very high to get to becoming more efficient. At all the major car manufacturers, they have invested a lot into getting more efficient engines. Of course, also some companies go toward fully electric cars. But still, as I mentioned, the major problem is batteries. If you have batteries with a higher energy density, you solve the problems immediately.
Thank you.
First of all, thanks for a nice lecture. I was just wondering what your opinions on things like proteorhodopsin or bacteriorhodopsin, which is a much simpler system in bacteria that's surely easier to manipulate. And do you think there's any potential to that, or?
There's no potential, because the quantum yield is pretty low. The quantum yield of bacteriorhodopsin is the order of 0.2 to 0.2%. Not 0.2 to 0.2 to 0.3.
But my point is
20 to 30%.
Sorry,
it's only a very limited range of waveflight, and it doesn't transfer electrons. You don't produce reducing power. So I don't think it's of any use for energy conversion.
But like my point was that because it's such a simple system that you can actually produce in mass, because
it's very easy to produce tons of bacteriorhodopsin. Very easy, but it's not much. There are a few sensors can be made of it, but not for energy conversion.
Thank you.
You indicated in one of your last slides that solar photovoltaic, they have around 20% efficiency in capturing the sun energy. Yeah. And also you talked about the farms that will be located in the Sahara, in Algeria or North Africa, and the Saudi desert, or the Taklamakan in China, or whatever. And companies like Desert Tech, which you indicated in your lecture, already have a project, I assume, in Algeria, I think.
Yep.
So it looks to me this is much more efficient than biofuel.
That's right.
I mean, when we compare the efficiency of between the different methods.
Yep.
So it looks like this is the way things are going. From your experience as a European, And maybe you're melting with the different project being studied in Europe or across the developed world. What is the possibility of this option of energy in comparison to other option of energy like biofuels or wind energy? Or thank you very much.
Of course, let me say wind energy. As soon as you have enough wind, it would be my preferred option. And actually, of course, there are regions in the world like Patagonia or the roaring 50s where you have lots of energy, and if you can produce lots of wind energy. And what is kind of not known in wind energy is that it goes with the tube of the wind speed. So if you double the wind speed, it's by a factor of 8, which you get more energy. Yeah? And so we have to put the we have to use the wind energy of the to make use of it in the areas where there is lots of wind. And if we could do maybe energy consuming processes like aluminium production into that areas, we could use the wind energy there, and the wind is always blowing the roaring 40s on Patagonia. So I think this could easily be done. So wind energy would be my preferred option. Next then, of course, would be photovoltaic cells. And in Germany, as I said, if the sun is shining full light, we are reaching very soon 100% of the consumption. But the major problem is that we cannot shut off the coal-driven power stations so fast. So this has to be run continuously, and in order to go on. In order to go on, also, of course, nuclear power stations cannot be switched off within a few minutes or within a few hours. So this also have to run for basic supply. For the moment, Germany is producing much more energy than we actually consume on warm, sunny days in summer. And we export the energy subsidized, and the system in Germany is like that the production of the production costs, or the costs for the consumer has to pay the guaranteed price to the producers of sun energy. This used to be very high, and this actually increased the price per kilowatt hour by 6 euro cents per kilowatt hour, which the general consumer has to pay. Even the cleaning lady or people with low income, it's not a government support, not a government subsidies for the support, but the support comes from the users of the electricity. And that's, in my opinion, is a bad system. And this energy actually then is used, and this actually brings down the price for the energy which is exported. So the problem is that the producer gets 30 euro cents per kilowatt hour, and a buyer in the Netherlands or in Poland has to pay 2 or 3 euro cents. And the difference is paid by the German consumer. And that's a system which, is, in my opinion, in long term is not viable. Very bad.
Okay, thank you.
Thanks for the nice talk. I have two simple questions to this repair mechanism you mentioned. First, does it play a role in the efficiency that you need energy to repair? And secondly, do you know approximately the number of cycles each sensor does before it breaks?
I have no ideas about it. I only can tell you that plant exchanges the D1 protein, takes apart the entire photosystem too, changes the D1 protein, which is the heart of the photosynthesis, which also carries the manganese cluster, and this is taken out and made by newly synthesized protein. But of course, you can calculate, the energy need for the biosynthesis of the protein, but I have no idea how much of the energy is actually consumed. But of course, you also need energy for DNA synthesis and other things, and protein synthesis in general, which is normally not considered in this kind of calculations.
Yeah, I was just curious why you didn't comment on nuclear power.
Nuclear power, in my opinion, should be used. Of course, I'm not in favor of this closing down all the nuclear power stations in Germany. And this was simply a measurement by our chancellor in order to be re-elected. That was the only thing. Just 1 year ahead of the polls, and it's a mistake. I don't think there would be no probability that a nuclear power station in Germany would explode.
Any other question? If there is no, I might ask you a question.
Yeah.
In France, there are a lot of biorefineries, and they are making a tremendous amount of money. I know they are making chemicals from biorefinery, but they use biomass a lot. Do you see any future for biorefineries? And
Yes.
my second question would be, Boeing was here not so long ago. They consider making 20% of biofuel for their plane. So they cannot ignore your strategy or your philosophy, so they don't care.
There's no chance to produce enough biofuels from biomass, in my opinion, with that respect. But also, one has to consider in Germany, we produce for the moment, 3 million tons of biodiesel every year. But if you do the calculations, how much of that could have been grown in Germany, it's maybe 20 or 30%. All the rest of the rapeseed has to be imported from Great Britain. They grow lots of rapeseed, and also from Poland and from Ukraine. So we already now, in order to provide 6% of biodiesel into the diesel, we have to import the rapeseeds, which doesn't make much sense for me. Of course, there are very powerful lobby organizations talking to the politicians, and they just want to make profits. And it's also very clear that the farmers in Germany got lots of profit. They double their income from the biofuel stuff. But it also means that the price of the food increased. And as I mentioned, we now import wheat, and we exported wheat 5 years ago. No, there are drastic economical consequences.
Okay. Yes, still another question?
So thank you for your presentation. It was really amazing. I have a question. You said that one of the problem in producing biofuels is the lack of water. So I'm curious, we have 2/3 of our surface are oceans, right? Yeah. So can we use seawater as a water for for the photosynthesis?
Because seawater, there are not many plants that can be grown with seawater. We cannot simply we don't even have enough area for growing plants with seawater. Of course, you can use a little bit of these mangroves in order to do that, but you cannot grow many plants in seawater. In order to use desalination, in order to get water for growing the plants would be economically a big loss. What's the cost for 1 cubic meter of water? I was told it's about $1, at least in Singapore. And if you need 6 cubic meters of water per liter of diesel, then of course already there you have a big economic loss of money if you do that.
Okay, so if there is no more question, we want to thank you very much for this very stimulating discussion.